Okay, so hello, welcome to this small tutorial video that covers a couple of papers that we published in 2020. So this is based on the talk that I did today on my second exam, and I wanted to record it there, but I didn't. So I'm doing the recording now, just here, just for the YouTube. So okay, so the talk is on the topic of symmetry-inspired analysis of biological networks. My name is Ian Leifer, and I'm currently a graduate student, and my advisor is Professor Mákse. So yeah, so I'm going to start with the introduction and talk about what, the research that we did. So in general, in science, one of the central themes is to decompose the complex system into the fundamental building blocks and study the way collective behavior emerges from interaction of these building blocks. And in biology, there are two approaches that are fairly popular. One approach is modularity by biological function. That is from experimental results. So the big network is taken, and then, from the experiment, eventually, we figure out how to say that, oh, these pathways are related to metabolism of this sugar, and then this part is, related to metabolism of another sugar. And then we can say, okay, let's separate this and consider them separately. So it's coming from experiment. And another approach that is fairly popular is network motifs. And I'm talking about approaches to decompose networks. So network motifs are graphlets, like small subgraphs that you can go and count how many those appear. And so you can find that some of them are statistically more abundant than others in certain networks rather than in random networks. And so you can make conclusions based on this. But the main problem with this is this is not theoretically principled approach. So here we offer a new theoretically principled approach that is based on symmetry fibrations. And this is the way to look for building blocks that originate from the synchronization in the network dynamics. So we find these building blocks that are based on synchronization. And fibrations in general initially have been introduced in category theory by the greatest mathematician of 20th century, Alexander Grothendieck, and in late 1950s. And they were later studied in computer science, chaos theory, graph theory, control theory, and all over the math and computer science, and providing us with really well-developed mathematical machinery to work with, so it can be really applied widely to these biological networks. And before I go into methods and all the things, I want to say a disclaimer that here we talk about applications to transcriptional regulatory networks, because this is really what we did in our paper. But this is not, and we also use examples from bacteria, but these methods are not limited to biological networks, but they just, this can really be applied to any directed network. So, just keep that in mind. Now I'm going to go into methods. So first slide on the method is on method of transcriptional, what is transcriptional regulatory network? Now, so there is a DNA sequence. So DNA sequence is there to encode genes. So let's consider how gene Y is producing the protein that it encodes. So genes are there to code the proteins. So gene Y first, so DNA opens up, kind of opens up like this, and then the sequence is copied. And then in the process called transcription, this sequence is copied on the outside, and then we get mRNA. Through the transcription, and now from mRNA, ribosome comes here to read this mRNA, and then the process called translation, the amino acid chain is formed. So slowly, like this, another ribosome comes, connects to the next one, and then slowly the long amino acid chain is formed. And now the next thing that happens, and this is called translation, and the next thing that happens with this long amino acid chain is protein folding. So this long amino acid chain folds into the protein. So now these genes produced, so using these information code in this gene, we produce the protein. Now, this protein can do two things. One thing is it can go into metabolism here, or alternatively, it can go into go back and regulate the network. So there is, for instance, this was gene Y and this is gene X, and maybe this gene X has binding sites. So binding sites are certain places on the DNA, certain sequences on the DNA where proteins can come and bind and change the expression of gene X. So in this case, green is activation, red is a repression. And let's say in this case, this protein can come back here and activate the repression of gene X, meaning increase the expression of gene X. And red ones, let's say, would be decreasing the expression of gene X. So this way we can say, okay, so there is a activation link between gene Y and gene X, and in links like this, we can build a bigger network. And and so this is the introduction to TRNs, transcriptional regulatory networks. Now, before I go into the rest, I also want to say that it's really important, the fact that we put together all these methods that are coming from all different theories, I think is a pretty great achievement, even disregarding the rest of the results, which are, I think are pretty good. But it's really important to, to put all these methods together. So I'm going to talk about the definitions that are used. So I'm going to start with the input trees. So first, let's consider this small network, which is coming from E. coli, from bacteria E. coli. And this is a transcriptional regulatory network of this bacteria, like a small piece of it. And here, black is the activator link, and red is repressor link. There are two types of links, and each node is a gene. And now let's consider the incoming edges of each node, or k-in, or input sets. So if you look at like red node buyer here, it receives from itself, and it receives from CPXR. So here you can see buyer receives from itself and from CPXR. Spy receives from buyer and CPXR. So here are also two things. CPXR receives from itself. Here CPXR. All these green nodes receive from the CPXR. So here it's easy to see. So these are incoming edges. But now we want to define input trees. So input trees are k-in of k-in of k-in of k-in taken infinite amount of times. So we start with this buyer receives from itself and CPXR, again, and this is going to happen until infinity. Now CPXR receives from itself, etc. So we build these input trees, and now similar way for spy also, from buyer and CPXR is here, CPXR itself, and you build these input trees, and here is CPXR from itself. So we build these input trees now. Now, the important thing to define here that are going to be used a bit later is branching ratio. So branching ratio is here. So we count how many nodes are on each layer of this input tree. So the first layer is one node, second layer is one, etc. And then we define here by a branching ratio as ai plus 1 over ai, so number of nodes in the next layer over this layer for I going to infinity, so for infinite depth. So here is obviously it's going to be 1. Here is also obviously going to be 1 because I plus 1 over I, for I going to infinity is going to go to 1. So it's input trees and branching ratio. And now I'm going to go into isomorphism and fibers, and this is all based on this paper that I mentioned from Grothendieck in late 50s, where this was applied to category theory, but then later it was applied specifically by Paolo Boldi and Sebastiano Vigna for fibrations for graphs specifically in early zeros. And we were the first ones to really apply these to some real biological networks, or frankly to any real networks. So no one did this before us. Now, as I mentioned before, so these are the input trees of the nodes here. So if we consider this small subnetwork of these three nodes, let's consider these two red nodes for a second. So we can't just change these two nodes, because if we change them, then the stealth loop is going to move here. Direction of this link is going to change here. So it doesn't seem like these two nodes are symmetric in any way. Now you can change this red node with this green node, obviously, too, because this big network is going to be affected if you change them. It's going to mess everything up. You can change these two for the same reason. And so it seems like these networks of these three nodes doesn't have any kind of symmetry in it. But actually, if you look at the input trees, you would see that input trees of these two red nodes have the same topology. And they have the same topology, or mathematically they're isomorphic. Here, isomorphism is shown, which is the morphism, the map from one tree to the other, from one graph to the other. Really, these are trees, but from one graph to the other. And so you have these isomorphisms here. So these input trees are isomorphic, have the same topology, and what this represents is it represents the same information that nodes receive from the network. So this is the symmetry in information that nodes receive. Now, if you consider the green nodes, they also have the same input, and the blue nodes also comparing to each other. Blue is different from green because there is this type of different type of link. Here is repression, here is activation. It's very different. Now, these are isomorphic input trees and fibers. So the next thing to talk about here is fibration, so symmetry fibration. So if we consider, and actually before we go there, so symmetry fibration is a map between two graphs that satisfies the lifting property. And lifting property is defined here. I don't want to define it here because I think it's mathematically like it's kind of hard to like just perceive it by listening. We do review it in our review paper and then a couple of other papers, so you can refer there. But here let's think of symmetry fibration as the following. Let's think of it as a map from the graph to the base of the graph. So this is called the base, so the target of the fibration. So fibration is the map between two graphs. So it takes this graph that is called universal total graph. Let's just refer to it as graph, and the base. So this is the target graph. And what it does is it takes all the nodes of the same color or all the nodes in the same fiber, and it puts them together. And then it takes, and it does it for all the fibers. So 2 red nodes are put in this one, all green nodes are put into this one, blue here into this one, Now, another example here, more elaborated to illustrate something is this one, where these 2 blue are collapsed into this one. So if there is external link going to this node, then it's protected here, preserved here. And then here, the same story, when red receives from this white, it's also preserved. So this is the fibration of this circuit. So yeah, now the next thing to talk about is, so, you know, we defined all these fibers, vibrations, you know, why all this complex mathematical machinery, you know, why do we even care? Well, 2 things. One thing is this is a pretty like novel representation of the network, where, you know, you represent the whole network is just, or actually like the part of the network that corresponds to a given node with this new representation where it's like shows the information that the node receives. So it's a very novel way to think about this. Oh, like, okay, this is what's available to this node, you know? But another way here is actually, let's remember for a second that network actually represents the system of ODEs. So for instance, if you have this system of ODEs here, this network represents it. So let's start with these ones, with the late ones. So all of them have the decay, okay? So dx over dt is equal to minus kx. This is the decay term. Let's just, you know, not talk about this too much. Just each node can have a decay, so can have some kind of function that and it doesn't have to be a decay. It can be any kind of function that depends on this variable. So, and this, by the way, is like k, just a random function for now. Now, and there are also coupling terms. So we want to look at z1, z2, for instance. They receive from y1. So there is a link from y1 to z1. Therefore, z1 over dt has the coupling term here, g, that depends on y1. And same story here, the z2 depends on y, also g on y of y1. And here we will look at x. So x here has the external function f of t. So x really interacts with the rest of the network, and we just want to, you know, model this with f of t without going deep and explaining, you know, what really is going on there. So we put this interaction here, and so this is the network that represents the system of ODEs. So since we consider transcriptional regulatory networks, and since we just want to look at one-dimensional case here, we just consider kx as a decay is minus alpha x, and gx we just consider this simple theta function, you know, with a couple of parameters. So that function just on and off 0 or 1. And I want to, before I go further, I want to say that these is done here just for simplicity. You know, it's not here to really, you know, explain anything. It can be much more complex. You know, each one can be a network of the each one can be a system of few ODEs. You know, and coupling can be very complex. It doesn't have to be a theta function. It's just here for simplicity. Now, what has been shown in recent 20 years in these few papers is that symmetry fibration, existence of symmetry fibration leads to synchronization. So if there is this network that can be collapsed into this one, this is an example from before. So there is a system of ODEs here that corresponds to the bigger network here. So if we take these 2 red nodes, let's call them x1, x2, you know, green nodes, let's call, just take 2, y1, y2, but really y1, y2, etc. And blue nodes, z1, z2, etc. So we can write this big system of ODEs that corresponds to this network, and we can write this smaller system of ODEs that corresponds to this network with parameters that are chosen properly. So we look at x's, take parameters alpha, gamma x1, and gamma y2, and here they're preserved. So this is the base. So, you know, all the parameters of the initial network are preserved. And so we have this system of ODEs that describes this collapsed network. Then actually, so if there is this vibration, then for every solution of these network of 3 equations, so let's say solution is x of t, y of t, z of t, exists a solution for the base, for the original graph, such that x1 of t is equal to x, x2 is also equal to x, y1 is equal to y, y2 is equal to y, so all y's are equal to y, and all z's are equal to z. So really what's happening is that we take these networks and we go and embed it back there, the solution to this network, we embed it back there, and then we copy all the diagonals. So really the reason this solution exists, and it's been shown, you know, in all these papers, and not only it's been shown, but you know, then the next question, okay, you know, this solution exists, but you know, who cares? Maybe it never happens. Well, it does happen, you know, for our type of equation here, it does happen, and it's been shown. And in this paper, they did it for neural networks, and you know, this is a big area of study. The question, you know, the questions of stability and the basin of attraction are, you know, interesting questions to talk about. I'll just say here that we did you know, run some simulations here. So we took this smaller subnetwork, we ran some simulations, and what we saw is that we start with 2 different initial states, and then eventually they stabilize into this synchronized solution for this gene turned on. So for theta function equal to 1 that corresponds to this gene, and then for off they still stay together. So synchronization continues, and they stay, they come to this synchronous solution and they stay in it. And another thing that we saw is that we actually saw this in experiment too. So we took this like small subnetwork of E. coli, and in this subnetwork we saw this synchronization. So let's say there is this fiber A here that has the same inputs, input trees, and corresponding correlation, like this is the high correlation here, and there is no correlation with the rest of it. So we did see this in experimental expression data in this paper. Now the next thing here is to how do we find the fibers? You know, this is cool. Now how do we find them? Well, I'm not gonna, you know, go deep into this, but really fibers are equivalent to balanced coloring, or specifically fibers that correspond to the minimal fibration, minimal surjective fibration. These fibers correspond to minimal balanced coloring, or minimal balanced coloring also corresponds to equitable partition, and this has been shown. And in 80s there have been a lot of algorithms how to find this minimal equitable partition, and here this specific one that we used is based on like a more recent paper where it was applied to balanced coloring, which wasn't the center of it, but you know, this is the paper that we used. And so it's based on this definition of input set color vector. And I'm going to define what is balanced coloring in a second. So let's start with what is input set color vector. So input set color vector of a node is a vector that has the length that is equal to current number of colors in a graph. And each entry counts how many nodes of which color are in the input set or in the k-in of this node. So we take the node 1. Node 1 receives from 2 and from 3. And note here that there is only one color in the graph right now, so it's going to be one-dimensional. So 1 receives from 2 and 3. So 1 has the value is 2. The input set color vector is one-dimensional. The value is 2 here. So 2 receives from 1 and 4. So also 2. 3 receives from 4 and 6. Also 2. 5 receives from 4. So only 1. 8 receives from nothing. So it's only 0. Now we found these input set color vectors. So what? So the balanced coloring is such a coloring that where nodes of the same color have same input set color vectors, and nodes of different color have different input set color vectors. So in this case, we see that there are different input set color vectors that correspond to the same color. So this is not good. So what's happening is that we do the refinement basing on this. So refinement here is to say, okay, all these nodes with input set color vector 2 is new color green, 1 is red, and 0 is blue. And we create new coloring, green, red, blue. And so we color this new graph here, and we find new input set color vectors. And what we see, for instance, is that input set color vectors of 1 and 2 are the same, but 3 and 4 are different here. If you look, they're three-dimensional now. So 3 receives from red and green, and 1 receives from 2 green. So they're different. And so we find these new input set color vectors, and we create new coloring based on that. So we created this new coloring. So we created a second partition, and the third one is we have five-dimensional input set color vectors. And in this five-dimensional case here, we see we see that it stays the same. So 1 and 2 were the same color. They have same 3 same vectors, 3 and 4 have same vectors, 5 is different, 6 and 7 is same, 8 is also different. So here we found the balance coloring where all the conditions are satisfied. So this is the basic idea for this algorithm. Now we actually did implement this, and it's available as a package for R. It's available here on my GitHub, so you can come and download it. And so this is as far as algorithm goes. So this concludes the section on methods, and now I'm going to go into section on the results. So first I'm going to start with the results that are based on building blocks. So I'll start with the definition of the building block. Sounds like a good place to start. So building block of a fiber is, number one, all genes in the fiber plus all regulators that are needed to establish the synchronization, which for now let's not go into depth about this. I will go into depth about this in a couple of slides, but for now let's think fibers and all the nodes that regulate these fibers. And the next step is if any node in the fiber belongs to strongly connected component, we include the shortest loop through this component back to the fiber. So this is the building block. Now, yeah, so we did go and apply these to the algorithm to find these building blocks to like about 400 networks in this paper. Some of them biological, some of them not biological. And we found all different topologies of building blocks. So what do we do with them? Well, it turned out actually we can classify them, and we can classify them using only 2 numbers. So what we do is we classify them by topology of their input trees. And we define these what we call fiber numbers. So there are 2 numbers, we denote them like this, N and L. So N is the branching ratio of the input tree. If it's finite, then it's equal to 0, so branching ratio that I mentioned before. And L is number of external regulators of the fiber, just this number. So these 2 numbers are enough to describe this network. Now let's look at some examples. So let's start with a simple one. So let's say we found this building block here that has only these 2 nodes that are regulated by this 1 node. So this is a small building block of 3 nodes. This is the input tree. So now the branching ratio is equal to 0. It's not really equal to 0, but we define it as equal to 0 when input tree is finite depth. So first number is 0, and is equal to 0. And the second number is 1 because there is 1 regulator. So 0, 1 is how we classify this circuit. So this is 2 nodes that have 1 input, and it doesn't have to be 2 nodes. It can be 15 nodes that have 1 input, as long as input tree has 1 regulator and it's finite depth. It's going to be 0, 1. Now the next case here is 0, 2 when there are 2 regulators here. So it's easy to see that the input trees are going to be the same. You're going to have 2 regulators. I mean, same for green nodes. They're going to have 2 regulators, and they're going to be finite depth. So 0, 2. And this actually is a famous bifan. This is a famous network motif. So we found it here. And again, I will note that number of nodes in the fiber can be different. So it can be, I don't know, 15 nodes, and they're all regulated by these 2, as long as they're all regulated by the same 2 nodes. So this is the extension of bifan. Now, the next thing here is a more complex case where there is a self loop. So let's say there were these 2 nodes, there was a self loop here and the link here. And what's important is that no one regulates these 2 anymore. So these 2 have no external regulators. They can regulate the network, but they don't receive anything from the network. So, and this is the building block that has no regulators. So L is equal to 0. And because of this self loop here, there is this infinite chain. So N is equal to 1. So the branching ratio is equal to 1. And therefore we get this 1, 0, 1, 0 circuit. Now the next thing is 1, 1, where there is 1 external regulator here. So there is this infinite chain again, and there is 1 regulator. And so this is 1, 1. And this building block we call FFF, or feed forward fiber. This circuit, I'm going to talk about this more later. There is going to be a chapter on the dynamics. And in terms of the dynamics, this is actually a very important circuit. So we're going to talk about this more. I just want to show it here. And one more thing I want to talk about here is the 2-1 circuit. Is here there is 1 regulator, feedback loops and back and forth links. And what happens with input tree is that it keeps branching every layer. So N is equal to 2, 1 regulator 1, and in general case N is equal to 3, and N is bigger than 3, and then there is L. So this is a more general case. Now, Fibonacci and composite fibers. So there are these more complex cases where in the building block we wanna include the nodes that if nodes in the fiber are part of the strongly connected component. So in this case, there is a strongly connected component of 2 nodes here. So this extra link is included, the link back. And what happens as a result of this link? Well, if you look at the input tree, green node receives from itself from 2 white nodes. So we get these 3 here, and now the next, again, the same story, but one of the white ones receives back from green. Here is easy to see, so it receives back from green. So here there is another tree embedded in there. And what's happening is that if we look at the degree sequence of this input tree, we will see that it's actually a Fibonacci sequence, which is interesting. So this is a Fibonacci sequence, and as a result, the branching ratio becomes a golden ratio. So the branching ratio is a golden ratio. There are 2 regulators. So this is the way how this Fibonacci fiber is defined. And in a more general case, when there is a longer delay here of 3 nodes, so the shortest loop maybe is 3 steps or 4 steps in this case, like 1, 2, 3, 4, and there will be a delay here of I-4. So this is a generalized Fibonacci sequence, and there are and the branching ratio is correspondent to that, and there are these general. It has been studied, so this is a well-known. These numbers are well-known. So yeah, and now this is the generalized Fibonacci sequence. So the one thing to note here is that this Fibonacci circuit, this specific one, has all these positive links and 1 negative. So all these links are activation and 1 repression. And this is one of the examples of the physical thing that is called the frustration. So frustration is a certain situation where physical system has certain tension, where it can't stay in one state. And I'm going to talk more about this later, but I just want to kind of say it now that this is the example of frustration where there is 1 link that is off. And this more general case where there is a length d of this loop, so there is a delay d. And another case here is composite fibers or multi-layered fibers. So if we consider these green nodes, they receive the input from this one, and this would be a fiber in itself, and that would be all great. But there is also here another node that also receives input from red, and they would be synchronized because of this next layer. So we have to include 1 more layer to explain why they're synchronized. And yeah, so here we have to find this, these bigger circuits, and what we see is that there is synchronization, and these nodes are actually far away from each other in the network, which is interesting. Now the input tree of the green node is just 1 red node here, and now the red node after that has this input tree of 1, 1. So this small input tree is 0, 1, and this one is 1, 1, and therefore we call this 0, 1 plus 1, 1. So this is a composite fiber, which is classified like this. Now, yeah, so this is as far as building block definitions. Now the next thing here is the landscape. So we did run this for a lot of biological networks, and we looked at the landscape. That is the distribution of N and L, like how these numbers are distributed over these networks, over these many networks. And so here, for example, we see that number L is the amount of blocks with N equals to 0, and increasing L is decreasing. And here there is a bump here. So again, N equals to 1, L equals to 0 is a bit higher than high L, N equals to 0. So there is this another bump where it's decreasing. And then here the important thing is that N equals to 0 and N equals to 1 is really enough to define most of the blocks that we found. So some of them are N bigger than 1, but there are almost none of them. So most of them are here. There are some multilayer and Fibonacci too. So now if we consider these bacteria here in networks that correspond to bacteria, so E. coli, Bacillus, Salmonella, we actually see that there is N equals to 0 and N equals to 1, and they're just decreasing as I showed before, and here the same story. And if we compare it with, let's say, mouse and human, we see that N equals to 1 is very low here. So our thought on this, and what we see is that there are more multilayer and Fibonacci, in this case than here. Maybe not here, but at least these two. And what we conclude is that this happens because there are longer loops rather than shorter loops. So what we see in E. coli and Bacillus, we only have this one short feedback loop, but what we see in human and mouse, these longer loops where there are longer delays, higher time scales. So this is just a more complex system. So that's how we make sense of it. Now, this is the landscape of building blocks. Now, the next thing I want to talk about is strongly connected components. So, and I didn't say this before, but we did consider a very big network that corresponds to the transcriptional regulatory network. Well, of equally, very big is, you know, is a careful word. About 1,000 nodes. It's like a decently sized network, and in this, you know, decently sized network, we were able to like identify like one small piece here that corresponds to this, you know, the strongly connected component of CRP and FIS, which are which regulate these, you know, these like small, you know, set of circuits around it. And similar story here with pH connected components. So, you know, a different connected component that regulates these nodes around it. So we're able to distinguish them. And now Fibration, what Fibrations can do is they collapse fibers. So rather than studying this big complex network here, we can study this smaller network, and we can just find the dynamics of this bigger network just by studying this smaller one. So we can study the function of the bigger network without worrying about all these extra nodes. So it's a big simplification. And here, you know, and here we go from, I don't know, maybe 50 nodes to, I don't know, maybe 20. And here is also, you know, a simplification, how we go from maybe, I don't know, 15, 20 nodes to maybe, you know, 10, 15. So there is a simplification here. So this is another result that we show. Now, the next thing I want to talk about is dynamical results. And so what is really going on in terms of the dynamics? So FFF, the one I mentioned before. So this is the building block FFF. And let's consider first SAT-FFF. So SAT is satisfied. I'm going to explain what is satisfied a bit later. Well, really, you know, satisfied means there is no frustration in this network. So there is no frustration. In here, everything is activation. So all the links are activation. We do consider X as just external signal because X interacts with the entire network, and X wouldn't, like, you know, we can just think, okay, sometimes it's 1, sometimes it's 0. So this is just a signal. And Y and Z we describe using differential equations from before. You know, with these theta functions, they're, you know, multiplied here. And so here we can see the dynamics of these that corresponds to these differential equations. So we start at different points here, and we see as before how they slowly get to synchronized state again. And X is turned on and off. You know, when it's off, it's easy to see it's just decaying to 0. When it's on, decaying to a different value, but that's the idea. So this is the synchronization in the FFF. And the next thing here is the analogy to electronic circuits. And so we will consider this one small network, or one small node from the genetic network. So one node with the self-repression. So this is the simplest example of frustration, where there is one node that can stay in the same state. Well, why? Okay, so why? In biology, it's very common to think about analogs to electronic circuits. So let's consider this NOT gate that just has like, let's say this Y has a state, and it just goes back to not this state all the time. And, you know, really in logic circuits, there should be some kind of delay here, you know, otherwise it wouldn't work. But, you know, just consider this for the sake of, you know, like abstraction to think about. And, you know, so the Boolean model here is Y at the moment T is equal to not Y at the moment T minus 1. And so if we start at moment 0 at 1, then next is going to be 0, then it's going to be 1, 0, 1. So it's going to oscillate, it's going to go up and down. So this system doesn't have a state where it's stable. So there is this frustration that creates tension in the system. And now we can actually see the same thing in genetic network. So here we look at the continuous model here. I'm going to say, you know, this is first order ODE. We can't have oscillations in first order ODE, it's obvious. But here, you know, we actually, this whole thing is an oversimplification, obviously. You know, the transcriptional regulatory network is in reality, there are like few processes inside. So we have to like really look at concentration of mRNA, concentration of protein, and all these things, and there should be like, you know, these delays that happen just because there are a few equations in there. But we don't want to, you know, make this complex. So the only way to make it complex here, we put the delay in this equation. So we take C at the moment T minus tau. So this is the delay that we put here. And there are a few papers where, I didn't put the reference here, but there is a paper where they actually look at the delay that happened as a result of, you know, these being few dimensional, and they do in fact find oscillations in all these cases with self-repression, as long as there is a frustration there. And so, yeah, and here we see that when theta function is equal to 0, then it's going to decay to 0, and when it's equal to 1, it's going to decay to a different position. So here, what we see is we see these exponents that are kind of glued together, up and down, up and down. So we do see the oscillations in this case here too. So what we see, so these oscillations tell us that these type of circuits play the role of the clock. And this is a one self-repression loop. It's a small circuit, but this circuit is not just one node. This is UNSAT-FFF, so this is another FFF, but it has the negative link here. So these are two repression loops, and this one is still activation, but it doesn't matter. And this is UNSAT-FFF, unsatisfied or frustrated. And the analog to this circuit is the Wheeler mirror circuit here on the right, that has this X as an input, and Z receives from X and from Y, and Y receives from itself and from X. And if you were to write the state, moment T is going to be of Y and Z is going to be equal to Y T minus 1 and X T minus 1. So there are two cases, and you can see these two are same, so synchronization is pretty obvious. Now, when XT minus 1 are 0 here on the left, it's easy to see that YT and ZT is always going to fall into 0. So it's going to, whatever it starts with, it's going to get to 0 in the next step. So here is exactly what we see. So we start with 1, 1, we go into zeros everywhere. If you look at the states of X, Y, and Z, and time is like we start with this one, we go here, we start with this one, we go here. So the same story. And here, if X is equal to 1 is more complex case. So now we consider time, is changing. So we start with 1, 0, Y is equal to 1, Z is equal to 0, and then we go into 0, 0, and then we go into 1, 1, 0, 0, 1, 1, 0, 0. So we get oscillations very similar to what we saw before with this. If we forgot about Z for a second, just look at Y, we would get oscillations as before, as long as X is turned on. And so here we see simulations. So these simulations are for a different model. This is not the delayed oscillation equation. Sorry, this is not the delayed differential equation. This is the continuous state discrete time model, where we do see the same story with synchronization and oscillations. And we did study the period amplitude relationship here in our paper. But yeah, pretty it can be shown in the delay ODEs too, which didn't show it here. So here we find these type of circuits that has synchronization and oscillation. So all these circuits that have oscillation play the role of the clock, and the circuits that I showed before play the role of just synchronization so far. Now, the next thing to consider here is the process of symmetry breaking. So the process of symmetry breaking is the process through which we find circuits that are don't play the role of synchronization anymore. They play the role, don't play the role of clock anymore. They play a different role. Now, which role? Okay, so let's start with this circuit. It's the chain, and so we first consider the base of this circuit, which is just one node with negative self loop. And now we consider the process of replication. So it replicates into two nodes that have this back and forth link, which can be collapsed back to this. But this is a symmetric circuit, and this is a toggle switch. So discovery of toggle switch that started the whole field of synthetic biology in early zeros. I think it's actually in 2000, starting with the famous paper there by James Collins. And so this is a toggle switch, which is the memory unit, which can be at the state 0 or 1. And and now how does it switch between states 0, 1 and 1, 0? Well, there should be some inputs. So we take this one and we break. So this is so far symmetric. Now we break the symmetry with some inputs, with set and reset inputs. And if set, well, and I'm going to talk about dynamics a little more later. So this is SR flip-flop. This is a famous SR flip-flop, which is the analog, I'm going to show it on the next slide, but yeah, the analog to this is SR flip-flop. And there's another variation of SR flip-flop that is slightly more complex, that has more inputs here and the clock. And here this is another variation. This is Fibonacci. So Fibonacci becomes this one, which is broken by J and K. So this is a JK flip-flop. So these are all memory units. So before we talked about clocks, and here these are memory circuits, and this is the functioning of this SR flip-flop, the one I mentioned before. So this is the biological circuit, is the electric circuit analogy. And so there are only two states allowed, 0, 1, and 1, 0. 0, 0 is not allowed, and 1, 1 is not allowed. And the switch between them happens by setting set to 1 and reset to 0, and set to 0 reset to 1. When they're both 1 or when they're both 0, there is no change in state. So this is how this system changes the state. So this is the storage of the memory. So we show that there are these circuits, that some of them are oscillations or clock, and some others are memory. And I'm not going to show anything here, but I'll just say that in the paper we looked for the both type of circuits, and we did find significant amount of them that is more significant than in random network with the same degree sequence in a bunch of networks that we looked at. So these are functional circuits that we have found in the real networks. So I want to make a conclusion here and just talk about proposed research. The research that is going to be, so this is is the slides from my second exam that is not happening right now, but it's happened today. So I'm going to talk about proposed research a little bit, because right now it's in the future. I hope by the time you're watching this, maybe it's in the present already published. But anyway, this is what I'm going to talk about here. So first, conclusions. So fibration symmetries provide a novel way to analyze biological or any directed networks. So this is the way that I presented, in these last, 45, 50 minutes. Then the next thing is symmetries of the network help uncovering new building blocks that are related to synchronization, and they're functional building blocks. And some of them, along with synchronization, play the role of clock, and some of them play the role of memory. And so these are, novel building blocks that actually have the functional, reasonable function, rather than just statistical abundance or any other thing that connects them together. So this is a theoretically principled strategy to search for computational building blocks in these networks. And I also want to say, due to all these research in last 60 years, there are a lot of algorithms there. So it's very heavily algorithmically supported and very theoretically principled. And for further reading, I refer you to two of our papers, one of PNAS 2020, and another one is PLoS Computational Biology, also 2020. So this is for further reading. And now the last thing I want to talk about is the proposed research. Is the research I'm going to do for my dissertation. So the first part is we studied this, what I showed you before. So this is a small subnetwork of E. coli, and this subnetwork is has this partial synchronization. So as I showed before. So we actually now want to go and study this for the entire network of E. coli and Bacillus, and whatever the expression data, where expression data is available. So we want to check if this is true and see if this can be confirmed by the experiment. This is the first side of this, and the second side of these is this. So far we talked about perfect symmetries, and perfect symmetries are great, but the problem is with perfect symmetries is that it has to be exact number of edges. But if you look at the real networks, for instance, here, you look at this real network of C. elegans, and before we talked about real networks too, but transcriptional regulatory networks are actually fairly precise, but there are a lot of networks that are not very precise. So if you look at this network of C. elegans here, you see, if you consider these brown nodes here, some of them have only one link, for instance. And if there was another link here, then they would be perfectly symmetric. And here, you see that they have two links, but some of them have one. And now if you look at this whole thing and, 1, 2, 3, 4, 5 nodes are missing one link. So 5 links are missing out of, 12 nodes, 24 links. So 5 links out of 24 are missing. And if we, fix them, then there's going to be a perfect symmetry. So this is a pseudo symmetry, and that's what we want to study. Now, it's obvious to, when you do it by eye, when you look at this smaller network, but we want to develop the algorithm to actually do this on a bigger scale and apply it and, see what conclusions we can get there. So this is another part of the proposed research. So this concludes my talk here. So thank you for your attention, and that's all I wanted to share. So thank you. Hope you found this helpful.